Hello, everyone, and welcome to Chapter 9, Microbial Control. I call it Part 1 because I see this chapter as part of a two-chapter unit. On this slide, you see more clearly what I consider to be the two chapters that make up this unit, Chapters 9 and 10. Chapter 9 is about controlling microbial growth in the environment. We'll define that in the next slide. Chapter 10 is about controlling microbial growth in the body. So that chapter is going to be about antimicrobial medications like antibiotics. Now, Chapter 9 is divided up into three parts. There's an introduction, that's the vocab dump. There is a section on various physical methods of control, and then a section on various chemical methods of control. This particular PowerPoint presentation is only slides 1 to 16 of the 90 slide PowerPoint, and it gets through a good part of the introduction, but not all of the introduction. You'll find the rest of the introduction in the next file that's online. As is customary when we begin a new topic, I like to answer the question, why do we care? Why do we have to learn this information? And I'm going to do so by defining how I'm going to be using the term environment in this chapter. So the environment is any place that we might be running into microorganisms that we don't wish to, whether it's a healthcare facility because we're trying to control for pathogens, it's in the microbiology lab where we're trying to keep pure cultures, in a food production facility because you don't want to introduce food spoilage, organisms or pathogens into the food product, and at home where again we wish to control for pathogens and spoilage organisms. Let's start with the definition of what is microbial control. Now you might be able to come up with the first bullet on your own, reduce or destroy undesirable microbes in an area, although you might have come up with the destroy part and not the reduce part. But if you look at the subsequent bullets, you will notice they are all about reducing, preventing, slowing down. So not about destroying, but simply buying us time to, let's say, finish the jug of milk in the refrigerator before it goes bad. Okay, let's start by looking at the vocabulary we're going to be using in this chapter. This is an overview slide where you see that there are two broad categories. There's sterilization, and then there's another category, disinfection, antisepsis, with several specific vocabulary words that fall into that second category. As you move through the next few slides, at the beginning of each slide, I recommend that you pause this audio part, read through the slide before you listen to my audio. I'm not going to be reading the whole slides for you. I'm going to be just commenting on them. And so you'll want to make sure that you read the slide before you hear my comments. So as I mentioned, pause the video and take a moment to read through these three definitions. I'm starting with sterilization because it is the most rigorous form of control. It is the destruction of all forms of microbial life. It does kill endospores. It does inactivate viruses. Interestingly, prions, those are those infectious proteinaceous particles, infectious proteins, they are not deactivated by our current processes of sterilization. So why is it that we can get away with having objects called sterile if there may be prions on them? And the answer to that question is that human diseases that are caused by prions are so rare that from a public health perspective, there's a very low risk in continuing with these processes for sterilization that don't in fact inactivate prions. I had a student one semester though who was a surgical tech, and she told a story of how they had a patient who had an infectious prion disease, and they ultimately had to incinerate all of the objects in the OR after working with that patient. There was no other means of sterilizing the operating room. The other thing is because sterilization kills all cells, it can't obviously be used on any object that you wish to survive, so it can never be used on our body. And then the other thing I would say, the adjective sterile, that's an absolute term, meaning something is either sterile or not. It can't be a little bit sterile, just like the computer is either on or off. It's not a little bit on. It's an absolute term. So really the only difference in the definition between sterilization and disinfection is that disinfection doesn't kill endospores. So it is a high level of control that destroys all bacterial vegetative cells. It destroys all viruses, but not endospores. Disinfection, because it usually uses harsh chemicals or very strong physical methods, is not used on living tissue, so it's usually used just on inanimate objects. So then what do we use on the body? So the chemicals that we use on the body for microbial control are antiseptics. So this is used on living tissue, usually the skin, right? As I mentioned, drugs that are taken internally will be in the next chapter. So things that we use on the skin are called antiseptics, and antisepsis is destroying vegetative pathogens on living tissues. So as I mentioned, there are 3 additional vocabulary words that are related to disinfection. The first is degerming, and the most important part to remember of this definition is that it's a mechanical process. It's really about scrubbing your skin, and the 2 examples are using an alcohol swab to scrub the skin prior to an injection, or washing your hands with soap. Yes, there are some chemicals there, the alcohol and the soap, but primarily the scrubbing action is what's removing the microorganisms. The next term, sanitization, also called sanitation in some textbooks. The important part to remember from this definition is that it refers to the public sphere. And so this is about reducing levels of pathogens to a safe level in the public. And while a public restroom is sanitized, and you might have gone into a hotel and seen the little strip over the toilet that says sanitized for your convenience, at home you disinfect your bathroom because home is a private place. The last vocabulary word here is pasteurization. That is, of course, named after Louis Pasteur, the father of microbiology. And we will be discussing methods of pasteurization in the physical methods of control section. As I mentioned in one of the opening slides, sometimes we really do want to kill microorganisms, and sometimes it's simply enough to inhibit their growth. And so here are 2 adjectives that we apply to typically chemical agents. That refers to whether or not they kill a microorganism or they just inhibit its growth. So notice the suffixes there, cytal and static. So cytal, kill, static is inhibit. And the thing to consider about a microbiostatic agent is that if you remove it, because you didn't kill the microorganism, growth might resume. And so the example here is the refrigerator. So if you've ever left something out of the refrigerator accidentally, you know that it might end up spoiling really quickly because any bacteria that were on the food grew quickly while they were out of the refrigerator. This slide has vocabulary words that you used in lab, and I'm going to explain why this slide is here on the next slide. So take a moment to review these vocabulary words before you move on to the next slide. So the reason I bring back the vocabulary words from lab, asepsis and aseptic technique, is that they resemble a little bit new vocabulary words from chapter 9 antisepsis, and antiseptic. So I want to make sure you're not confused about the definitions of those 4 vocabulary words. Make sure that you know the differences between them. And I also want to make sure that you understand the differences and the similarities between disinfecting and sanitizing. So go back and review all the vocab words to make sure you're comfortable with them. And here is the obligatory everything you need to know in table format, at least when it comes to the terminology of microbial control. The key to this question is seeing the words antimicrobial cleaner. So that means like a hand sanitizer or a hand soap with an antibiotic in it. Because it has that additional chemical, the correct answer to this question is C, antisepsis. It's being used on living tissue, and it actually has an agent in there that is killing the microorganisms. If I had asked the question, if you wash your hands using plain old soap, you are performing, then the answer would have been E, degerming, because plain old soap doesn't have an antimicrobial medication added to it, and so it's just considered degerming. So if you see a question like this about washing with a hand soap, make sure to differentiate between one of those antimicrobial cleaners like hand sanitizer or soap with antibiotic added, and just plain old soap. This last slide is actually a table of contents slide for the entire chapter in a visual format. So at the top you see microbial control methods, and on the left in pink are all of the physical agents, and we're going to look at pretty much all of those. What I like about this image is that it tells you if the particular method sterilizes or simply disinfects. Down the middle in blue, the chemical agents, we're going to look at those, and again, some sterilize, some disinfect. That yellow square, chemotherapy, that's chapter 10. Chemotherapy originally meant any antimicrobial medication not specifically related to cancer treatment. We'll also look at a couple of mechanical removal methods. The one part of this that we won't cover are biological agents.